Okay, we're going to look at one of the specific examples in the syllabus, which is insulin, and how insulin triggers signaling pathways in the target cells. Okay, but I will be quite fast. I'll go quite fast for the examples, right, with insulin and glucagon, because we should be not so focused on all these details, right? As I mentioned in the previous videos, right, the most important is to understand the general stages of signaling and how signal is transduced, how the signals is passed into the cell, and then trigger the cellular response. The general stages is more important. The insulin and glucagon here serves as two more well-known examples for us to practice application of the general stages, and you need to know how to apply, right? Because again, I mentioned before, right, this topic, a lot of times the questions will involve not insulin and glucagon. They will involve other signal molecules and other pathways that we have not seen before, right? So in this part of the notes and in this part of the lecture, right, the lesson, I would hope that you approach it in this way. It's not so much of you remembering all the details of insulin and glucagon, but using the insulin and glucagon as practice, right, to apply the general stages. That's why I go quite fast, because I will assume that you understood the general stages already, right? So here I'll just brief through the specific things that's going to happen for insulin and glucagon, right? And again, you should be looking at all these details in the light of the general stages, right? So just take insulin and glucagon as any other case studies and practice the application, right? But guided with the details given to you, right, in the notes and in the lesson, so that you are more accustomed to apply the same general stages for other signaling molecules and other pathways with lesser details given. Okay, with that, let's look at the receptor. Right, so insulin receptor, right, insulin binds to insulin receptor, obviously. Insulin receptor is an example of a class of cell surface receptor known as receptor tyrosine kinase or tyrosine kinase receptor, right? So two names, tyrosine kinase receptor or receptor tyrosine kinase, or both are the same. So there are many different receptors that are classified as tyrosine kinase receptor, one of which is insulin receptor, right? So insulin receptor binds specifically to insulin binds specifically to insulin receptor, right? So this is how a general tyrosine kinase receptor looks like, right? Usually, right, for most tyrosine kinase receptor, it's going to exist as two separate subunits, and they dimerize, and then as a result becomes activated when the ligand or signaling molecule binds. Right, however, right, a bit of details for the insulin receptor. Insulin receptor does not undergo this dimerization because they are always a dimer, whether it is activated or not. Right, it's a disulfide linked dimer, right, disulfide bond, right? So it's a covalent bond, strong bond, right? So the two subunits always stay together for the insulin receptor. So there's no dimerization, there's only the conformation change for activation upon binding of the insulin for insulin receptor. But for the other tyrosine kinase receptor, for other cellular molecules, right, a lot of them undergo this dimerization. Okay? Right, so this is, a bit more. So just talk a bit more about the structure of the receptor, right? As a receptor, it's obviously a transmembrane protein, right? So it spans the entire width of the membrane. There is the transmembrane domain here, made up of usually alpha helices with hydrophobic R groups pointing towards the outside to interact with the hydrophobic fatty acid tails of the phospholipids. You learn this when you learn membrane. Right, that's the cytoplasmic or intracellular domain. Sometimes it's called cytoplasmic tail because it looks kind of elongated lah. So again, different terms for the same thing lah. So intracellular domain, cytoplasmic tail, talk about the same thing. All right. The special thing about the tyrosine kinase receptor is that the cytoplasmic domain, right, contains quite a number of tyrosine residues or tyrosine amino acids. Thus the name tyrosine kinase, which is meaning that the tyrosine in the receptor, right, in the cytoplasmic domain of the receptor will get phosphorylated because kinase, right, and it's phosphorylated by itself, right. There's going to be a tyrosine kinase domain, or a part of the receptor is actually having an enzymatic function, right, tyrosine kinase function. And obviously this tyrosine kinase domain of the receptor is going to phosphorylate the tyrosine, right. Then the extracellular domain, right, would contain the ligand binding site lah. So for insulin receptor, it will contain the insulin binding site. Okay, so yeah, that's roughly the structure of a tyrosine kinase receptor, right? Which one example is the insulin receptor. Okay.